Okay, now we're going to talk about what happens to glucose and the start of metabolism. And really, we think of the start of glucose metabolism with the process of glycolysis. Okay, that's what glycolysis means. Is we're going to lyse glucose, break it into pieces, and in the process of breaking it into pieces, we're going to gain energy from it. And so another way to think of this is glycolysis is a set of 10 reactions where we're converting glucose to this molecule called pyruvate. So we're going to try to figure out how we do that and what's the point of breaking down glucose to pyruvate. So we know what glucose looks like, right? It's cyclized in the bloodstream as the alpha or beta enantiomer, and it can be polymerized to be stored as either starch or glycogen with our alpha 1,4 linkages, or in plants it can be stored in the beta 1,4 linkages and cellulose. So how do we get that glucose to start up glycolysis in our body from something like glycogen? Well, we undergo a process called glycogenolysis, right? We're lysing glycogen here, turning polymerized glucose into free glucose that we can use. Now, glycogenolysis occurs mostly in the liver and the muscles, which is where most of our glycogen is stored. And most of this free glucose is then transported throughout the body to be used to generate energy, and in particular to your brain, right? Your brain is a real big user of glucose. It prefers glucose as its food source, unlike things like muscles, which really prefer things like fatty acids as its food source. Okay, so we're going to use glycogenolysis as a small metabolic pathway to think about enzyme names and to think about metabolic pathways in general. Okay, so how does glycogenolysis work? It works by the action of these three enzymes. And let's look at the names of these enzymes because enzyme names typically give you a clue as to what they're doing. So the first enzyme in glycogenolysis is glycogen phosphorylase. Now the last part of the enzyme name, in this case phosphorylase, describes what that enzyme does chemically. And so glycogen phosphorylase is going to add a phosphate group to glycogen. The first part of the enzyme name is usually describing the substrate. So what does this enzyme do? It phosphorylates things. What does it do it on? It substrates glycogen. Okay. So if we think back to glycogen here, glycogen phosphorylase enzyme is going to phosphorylate this bridging oxygen and break it into two pieces. Now, where does that phosphate end up? Does it end up on the 4 position or the 1 position? Well, we'll have to figure that out. So let's look. The first enzyme, glycogen phosphorylase, breaks it into 2 pieces by adding a phosphate. Phosphoglucomutase mutases the phosphoglucose that we've just made. So what the heck is a mutase? Well, a mutase is a type of enzyme that moves one part of a molecule to another location. And in this case, it's going to move that phosphate group, which ended up on the 1 position after glycogen phosphorylase, and it's going to move that phosphate group to the 6th position. Okay. Now, the final enzyme in this metabolic pathway is glucose 6-phosphatase. This is a tissue-specific enzyme present only in the liver, primarily in the liver. And what does it do? It's a phosphatase, which is sort of the opposite of a phosphorylase. This removes the phosphate from glucose 6-phosphate. Okay? And the end result of that is free glucose. Okay, so when we think of this, we start off with glycogen, bound together with our alpha 1,4 linkages. We break that alpha 1,4 linkage by phosphorylating it in the middle of that bridging oxygen. That results in giving us glucose 1-phosphate as a product. Phosphoglucomutase acts upon glucose 1-phosphate and moves that phosphate group to the 6th position to give us glucose 6-phosphate. And then glucose 6-phosphatase removes the phosphate, giving us free glucose as a product that can go into glycolysis. Okay. Now, what is glycolysis? Glycolysis is a cytoplasmic set of reactions. We have 10 reactions that are part of glycolysis. We start off with glucose, a 6-carbon sugar, and we end up at the end with 2 pyruvates. Pyruvate is a 3-carbon sugar. So we're not losing any carbons as part of this process. We're just lysing glucose into 2 pieces. And what else are we doing to it chemically? Well, if we look at glucose up here, it's got a lot of alcohol groups attached to it. And we look at pyruvate at the end, it's got a lot of carbonyls attached to it. So we are lysing glucose, and we're also oxidizing glucose. Now, what are the big products of this reaction? As we convert glucose to 2 molecules of pyruvate, well, there's lots of intermediates here. All these molecules are part of glycolysis, but they're consumed or destroyed as we move down our 10 steps here. So are there any real products from this? Well, here we can look and we can see, well, we're using an ATP here. We're using another ATP here. We're making 2 ATPs down in step 7. We're making another 2 ATPs down in step 10. So if we think of ATPs, which seems to be a big thing we want from metabolism, we're using 2, we're gaining 4, and so the net ATP production as we convert 1 glucose to 2 pyruvates is 2 ATPs. What else are we generating? A couple waters and 2 of these reduced cofactors called NADHs. NADH is a reduced cofactor, which means it's holding on to electrons. And that makes a lot of sense because we said, well, we're oxidizing the carbon chain of glucose to form pyruvate. When we oxidize that, we're reducing NAD plus to form NADH. And these reduced cofactors hold on to electrons, and we'll see later, if we have oxygen, we'll be able to harvest that electronic energy to form ATPs. Okay, so we've got 10 reactions here. We are generating 2 moles of ATP for every mole of glucose that we consume, and we're also generating 2 moles of NADH for every mole of glucose that we consume also. It's sometimes convenient to sort of break these 10 reactions into 2 pieces. Reactions 1 through 5 we can think of as this energy investment phase because we're consuming ATP. And then the second half of this metabolic pathway, reaction 6 through 10, is the energy payoff phase because we're generating ATPs and we're generating this useful reduced cofactor. So what does glycolysis do? Well, we're going to take the 6 carbons of glucose, turn it into 2 3-carbon pieces of pyruvate. We're going to add, phosphate groups to glucose, convert those phosphorylated glucoses into ATPs, and then ATP is going to be used in our body to couple with other reactions to drive biochemistry forward. Okay. So we've got our energy investment phase, where we're generating 2 glyceraldehyde 3-phosphate molecules, and it costs us 2 ATPs. And then we've got the energy payoff phase, reactions 6 through 10, where we're generating our 4 ATPs and 2 pyruvates as our final products.